Can you identify this fruit? Golden yellow in color. And this is Baccaurea Ramiflora. You can see the plant here. The tree. It is a medium-sized evergreen tree. You can have a close look here. You can see the Baccaurea Ramiflora, commonly known as Burmese grape. And why the Baccaurea? Bacca and aurea. Bacca means it is a fruit and aurea means golden yellow. So this fruit is a golden yellow fruit. Here we can see this Ramiflora. You can see Rami means branches and flora means flower. Flowers will be produced means if we can see the flowers in the branches. So that's why the Baccaurea Ramiflora is came, and you can see the golden yellow fruit. So now I am tasting one fruit. Yeah, here we can see the even fruit size is also comparatively very bigger compared to the some other places also. Fruit is very tasty, and the seeds is also smaller one. Medium sized evergreen tree. It will be found in the Southeast Asia. It is distributed in Indonesia, Thailand, China, Myanmar, and in India we can find it mainly in the northeastern states. This particular species is dioecious in nature, where the both male and female flowers are present in different, where we can see the fruit, a golden yellow fruits on the branches. And I am taking you to the one more tree where you cannot find any fruits. Yeah, meters only distance apart. This is a male tree. We cannot find any fruiting in this particular tree species. You can see on the branches there is no fruit, but just 5 meters here you can see on the branches there are very good fruit setting, and the help of that very near a tree which is very less than 5 meters, you can see the surrounding also. This is very pristine and it will be grown in a very moist condition. Moisture plays a very important role, and the seed is recalcitrant in nature, and we having a very less period of viability. It may be 15 days to the maximum 15 days, or sometimes even if we kept in a very good condition, the longevity of the seed can be extended more than 15 days also, and even few months also we can store this particular seed species. One work is done on the same genus other species, Baccaurea quartalensis. They have prolonged the seed storage and seed viability also by storing in the appropriate. The easiest method to propagate this particular species is just soaking the seeds in the 24 hours of in the water. Simple water soaking of 24 hours is good for the propagation of this particular species. Apart from this, the cuttings can be used. If the softwood cuttings is there, then the 3 nodal softwood cuttings can be taken and propagated. And even the IBA 1000 ppm and NAA 500 ppm is also very much helpful in the root initiation also. And we can try the hardwood cuttings, but when we are taking the hardwood cuttings, the thicker diameter one will roots very faster than the thinner diameter one. And apart from this, we also tried the different grafting technique. Cleft grafting is very much successful, and apart from the cleft, we tried cup and lip method and side veneer grafting also. Side veneer grafting is also advantageous, but apart from all the cleft and this cup and lip, then side veneer, cleft grafting is the very much beneficial one, and it will the success rate is also very much higher in this. And if you want to propagate and initiate the root, then sand is the best media to initiate the root. Just keep it for the 1 month. After the initiation of the roots, you try to transfer into the whatever the soil it may be. But my suggestion is vermicompost based media can be enhanced the seedling growth and vigor also. So soil, sand, vermicompost in 2:1 ratio resulted a higher rooting and success rate. So you can also try. So next, if you need any seed material also, propagation material also, you can contact me and grow this very unique fruit and very tasty. This particular fruit can be utilized for making juice also. And so in the Meghalaya, we can see evergreen forest. We can see this particular tree species. Here the local people known as Garo people, they consume raw, and they are also used for other purpose also. It is a widely grown and commonly distributed in this particular area. But in South India, Baccaurea quartalensis is highly distributed. Ramiflora is not that much common. And Baccaurea cautelensis is very much common in the Western Ghats. Even it can be found in the Kerala also, and few studies has been conducted in the Kerala, and seed germination studies are has been done in the West Bengal and Meghalaya. We need to find it out the proper branches. It can be utilized for the. We need to find it out the appropriate diameter the branches for root initiation. So now I am considering this one, this branches. So what I need to do means I need to make a cut. So this one also I am doing 1 inch. Okay. Yeah. You can see. Zoom. Here you can see the layering cup. So this is the plastic one, and if you don't have any this kind of a layering cup, then simply you just take the polythene cover and you can put the growing media. Along with the different growth hormones IBA, NAA and IAA, I am also trying with a different effect of different growing media. So I have selected sand and cocoa peat. Why I selected sand means in the initial experiment of the cutting experiment, we found the very good results from the sand. It enhanced the root. So that's why instead of keeping the simple sand to absorb the good moisture, to maintain the moisture condition for the longer period, we have selected cocoa peat also. So cocoa peat and sand 1:1 ratio we have selected, and apart from that a simple sand, soil sand and soil cocoa peat also we are trying. So totally we have taken the 3 growing media. The moistened cocoa peat and the moistened sand is mixed 1:1 ratio. And so even in the rooting experiment of cuttings, layering, growth hormones can be applied either in the liquid form also, or you can apply in the powder form also. So for cuttings, quick dip method is there, or you can put in the overnight in the solutions. So in the layering experiment we have taken the growth regulators, and one thing is very important, you need to maintain the homogeneity in the experimental different treatments and replications also. So whatever the quantity, how much quantity you are applying for each the unit, you need to take care of that one. So applying more quantity, applying less is not good. So we need to maintain the homogeneity. So we have to apply this the powder. Along with the talc powder, I have prepared the different ppm solutions. So I am applying this one now. Yeah. After applying this, putting this layering cup here. So after applying, just one clip is there. You need to, yes. This is a very simple and easy technique of layering. Later, if you found the moisture is very less, then you can put the water from here.